Alright, so in this video we're going to be looking at mitotic division, and this is how eukaryotic cells divide. Now if you remember from earlier, prokaryotic cells divide through binary fission, which is a relatively simple process because those cells are simple. But eukaryotic cells are much more complex, so their method of division, mitosis, is also complex and involves multiple stages. So let's break it down. There are four main phases of mitosis, and a really common way to remember this is the acronym PMAT: prophase, metaphase, anaphase, and telophase. So let's go through each one. First of all, we've got prophase. Before mitosis begins, the DNA has already replicated, so the cell now has two copies of its DNA. At this stage, the DNA is in a loose thread-like form called chromatin. During prophase, each chromatin condenses into chromosomes, which are much more compact. Each chromosome consists of two identical sister chromatids joined at the centromere. Think of it like packing a suitcase. If your clothes are all loose and messy, it's hard to move them. But once everything is neatly folded and packed, it's much easier to transport. The cells are doing the same thing with its DNA. Also during this stage, the nuclear membrane breaks down and the centrioles move to the opposite poles of the cell. Next, we've got metaphase. Here, spindle fibers attach to the chromosome and line them up in the middle of the cell. Think of it like lining people in the middle of the center before splitting them into two equal teams. Everything has to be perfectly organized before the division happens. This is a really important checkpoint because if things aren't lined up properly, the division can go wrong. Then we move on to anaphase. This is where the sister chromatids are pulled apart and moved to opposite ends of the cell. You can think of it like a tug of war where each side pulls one copy away so that both get an equal share. Finally, we have telophase, and at this stage, a new nuclear membrane forms around each set of chromosomes, and the chromosomes begin to unpack back into their loose chromatin form. It's like once everything has been delivered, you unpack your suitcase and you spread things out again so they can be used. And after this, the cell undergoes cytokinesis, where it fully splits into two identical daughter cells. So overall, mitosis is a highly organized process that ensures each new cell gets an identical copy of its DNA, kind of like making a duplicate of the original cell.
Now let's quickly test your understanding. So pause this video and have a think about this question. Now let's go through it. Before mitosis, DNA is replicated, creating two identical copies. During mitosis, these copies are evenly separated into the two cells. This means that each daughter cell receives the same genetic information. So the key idea here is the replication and also the equal separation ensures that we get those identical cells.